So this journal art. Hello everyone, my name is Amanda Trish, and this is entry 2, Genomic Rearrangements and Genetic Disease. So this journal article that had a news article published in Science Daily delves into the intricate nature of complex genomic rearrangements or CGRs and their significant implications for structural variation within the human genome. As an overview of complex genomic rearrangements, CGRs are structural variations that result from alterations in the normal arrangement of genetic material within chromosomes. These arrangements can manifest as duplications, deletions, inversions, or translocations, often resulting in altered gene dosage or disrupted gene function. The study focuses on inverted duplication-triplication structures, which occur when a segment of DNA is duplicated and inverted, contributing to a complex genomic landscape. For methodology, the researchers employed a multifaceted approach to analyze genomic rearrangements in a cohort of 24 individuals, integrating technologies such as high-resolution genomic sequencing, optical genome mapping, and CRISPR. For high-resolution genomic sequencing, this technique provides an in-depth analysis of the genetic code, allowing researchers to identify single nucleotide variations, or SNVs, insertions, deletions, and complex rearrangements at a level of detail. By generating vast amounts of sequencing data, high-resolution genomic sequencing enables the detection of subtle changes in the DNA that may contribute to various genetic disorders or phenotypic variations. For optical genome mapping, it complements genomic sequencing by offering a visual representation of the genome. This technology utilizes fluorescently labeled DNA molecules that are stretched on the surface to create a physical map of the genome. The resulting images provide a unique perspective on the structure of chromosomes, allowing for the identification of large structural variants. OGM is particularly valuable for detecting complex rearrangements that may be missed by sequencing alone, as it allows researchers to visualize the spatial arrangement of genomic regions. For CRISPR, they used it to target modifications in the genomes of these subjects to investigate the functional consequences of genomic rearrangements. By knocking out or modifying genes of interest, the scientists can then assess how these changes can impact cellular processes, developmental pathways, and overall phenotype. For implications of gene dosage and disease, the formation of inverted duplication is primarily attributed to the template switching during DNA replication. This phenomenon occurs when the DNA replication machinery inadvertently switches templates, especially at low copy repeats or LCRs, which serve as hotspots for recombination. LCRs are regions in the genome that consist of repetitive sequences, making them susceptible to errors during replication and recombination. The study reveals that these complex rearrangements have profound implications by directly influencing the expression levels of dosage-sensitive genes. For instance, disorders such as MECP2 duplication syndrome arise when there are alterations in the number of gene copies, leading to an imbalance in gene expression, which disrupt normal developmental processes and contribute to the onset of neurodevelopmental diseases. Contributions to genomic instability. The researchers provide new insights into the mechanisms driving genomic instability, specifically through non-allelic homologous recombination, or NAHR, and replication fork stalling. NAHR occurs when non-homologous chromosome regions mispair, leading to structural variations. Replication fork stalling, on the other hand, can create areas of tension in the DNA, increasing the likelihood of template switching and subsequent rearrangements. These processes create complex structural haplotypes, contributing to variability in gene dosage and influencing the severity and heterogeneity of associated diseases. This demonstrates the evolutionary role of structural variants and connects with class topics on how duplications drive both innovation and genetic disorders. Thank you.